Hi, my name is Sarah Saldana, and today's date is January 16th, 2018, and in this video I will be demonstrating aseptic techniques for culturing microbes. So I went ahead and I already did my E. coli culture. I'm just going to go ahead and show the process. I have my materials here, like my cup with alcohol, in order to sterilize the pipette that I used, which is right here. I'll go ahead and show you my original nutrient broth, the vial of the E. coli culture that I just used. I have a little more. The candle here, which I used to sterilize both the lip of the nutrient broth and the E. coli. So basically what I did was, after sterilizing this pipette and sterilizing both the lip of the nutrient broth and the E. coli, I went ahead and I put 0.25 milliliters of the E. coli or the nutrient broth inside of the vial containing the E. coli culture. After that, I shook it until the whole tablet dissolved, and then I placed all of this solution into the nutrient broth here, which I previously already labeled E. coli. But before capping it again, I went ahead and I sterilized the lips to make sure that no other microorganisms in the air or any from my glove that, I made sure didn't touch it, but if any had touched it, we went ahead and killed it so it wouldn't interfere with the E. culture, which is now inside of here. And this is the procedure that I'll be repeating for the S. epidermis and the S. cerevisiae culture that I have. Later, I will incubate the broths upright and untouched for 48 hours in an already previously selected area. And developed cultures will either appear cloudy or show a flocculent growth at the bottom of the tube. However, this is the end of exercise 1 for aseptic technique culturing microbes. Hi, my name is Sarah Saldana, and today's date is January 18th, 2018. And this video is going to be exercise 2 of the aseptic technique and culturing microbes lab for isolating individual colonies. So, I'm going to go ahead. I have my S. epidermis broth right here. I have my agar plate, the corresponding one that's already been labeled. I have my candle, and I have my inoculation loop in my cup of alcohol. So, I'm going to lower this so we can go ahead and get started. First thing I'm going to do, I'm going to try and light this candle. There we go. Just going to be used to sterilize the lip of this broth. So I can go ahead and take out my inoculation loop from the alcohol. Removing the cap, I go ahead and run it through the flame to go ahead and sterilize the lip. Go all the way around. And I'm going to be very careful to make sure that my gloves do not touch the now sterile lip. And I'm going to place my inoculation loop inside so I can get some of this S. epidermis. I'm going to move this flame out of here. And what I'm going to be doing is I'm going to be in zigzag formation, placing this inoculation loop into quadrant 1. This here agar plate. So we go zigzag. Perfect. I can recap that. Putting the inoculation loop back into the alcohol. Now before I cap the S. epidermis again, I'm going to go ahead and run it through the flame just to sterilize. Make sure my glove did not touch what needed to stay sterile. There we go. I can go ahead and now place this to the side for future labs. Could put that out, we don't need that anymore. Alright, and now I'll go ahead and take the inoculation loop again out of the alcohol, and we're going to move some of quadrant 1 to a zigzag formation into quadrant 2. Place the cap back on that. This time I'm going to leave the inoculation loop inside of the alcohol for about 20 seconds before going ahead, and I'm pretty much just going to go ahead and repeat the same process there for quadrants 3 and 4. So Now we can go ahead, and I'm kind of just turning the agar plate just so I know which quadrant I just worked on and which one I need to. So working on the adjacent one, I went ahead and did the top, I did the left, and this next one now, I'll be going from quadrant 2 into quadrant 3. There we go. Putting that lid back on, rotating it just so I don't mess up which quadrants I'm working on. Now I've already done one other broth before this video, so all I have left now is the E. coli, which I'll pretty much be doing the same steps I just did for the epidermis, and then I will go ahead and leave the agar plates to incubate for another 48 hours before being able to see the individual isolated colonies. So, for another 48 hours. Hi, my name is Sarah Saldana, and today's date is January 20th. And it's been 48 hours since I used the inoculating loop to go ahead and spread 3 separate bacterias onto 3 separate agar plates. So this was in order to isolate individual colonies and check for microbial growth, and I'm kind of disappointed because the only one that looks like there was real growth that's noticeable is the S. cerevisiae. So I'll go ahead and show. We can see there's a little bit of growth in that top quadrant. But not so much anywhere else. I mean, I know the top quadrant was the one I started with, and then I went counterclockwise, but there's really nothing in the other areas. So there's that one. Again, here's the S. epidermis, and there's not much in any of the quadrants. There's no growth of any microorganisms. And now here is the E. coli sample. Again, this would be the top quadrant, so it should have been the strongest quadrant with the most microbes, but there's nothing. So what I'm going to do is I'm going to go ahead and I'm going to try letting it incubate for another 24 hours. I'll check on it tomorrow, and if I see any differences, I'll go ahead and update this video, but so far the only one that I was able to tell, the difference between isolating the 4 quadrants was, again, the sample S. cerevisiae. So that's that. Hi, my name is Sarah Saldana, and today's date is January 21st, 2018, and I am just updating. It's been another 24 hours of incubating the 3 different bacterias, the S. cerevisiae, the E. coli, and the S. epidermis, and still, unfortunately, there is no growth of either bacteria seen in any of these quadrants, not even the first, which should have been the strongest for the streak plate method. However, there was some more bacterial growth in that first quadrant for the S. cerevisiae bacteria. So again, in my reflection, I wrote maybe, one of the reasons is because when I was pouring the agar plates, I did do it just an hour before starting the experiment. Perhaps in the future, I'll be sure to pour earlier just to make sure the plates are really ready. I made sure that when they were inverted, the agar didn't move, so I assumed they were ready. And I was able to see microbial growth in this bacteria. However, I'm not too sure why the other 2 didn't change, but I just wanted to go ahead and update. Even after another 24 hours of incubation, there was no growth in 2 out of the 3 samples.